Sorry. So thank you for the introduction. Can you see the screen and hear my voice?
Yes.
Okay, perfect. And I found that there are a lot of things in between. I hope that will not be too annoying. Okay, so I will just start. So I'm happy to share our recent publication here and this symposium. My name is Wei-Ting Chen from Professor Bart De Strooper's lab in Belgium. So actually my work is not really focusing on the oligogenesis side, but in this presentation, you will hear how we use spatial transcriptomic and in situ sequencing to unbiasedly discover a coordinated network among multiple cell types, including oligodendrocyte, their response to a certain microenvironment, the pathogenic hallmark of Alzheimer's disease. So we know that Alzheimer's disease, which is a major type of the dementia. And in these images, this is the self-portrait of an artist, William, after diagnosis with Alzheimer's disease. So clearly it's a disease which will cause gradually cognitive dysfunction and eventually death. So the central hypothesis of the disease, and there are a lot of strong genetic evidence showing that A-beta is, there might be a very strong etiology of the disease. However, there are several failures of the clinical trial. The anti-amyloid therapy that can actually remove the amyloid plaque from the human brain, but they cannot really stop the disease progression. So they really make the whole field really hesitate. Should we still keep focusing on the A-beta? So in our working hypothesis, we have slightly modified this hypothesis. We still consider A-beta as a major trigger. It's important for the disease onset, but it's not eventually the driver of the disease, which means that A-beta overexpression or misfolding and the accumulated deposit in the brain, and that might induce abnormal cellular network around the plaque. And this cellular network that might be eventually independent of the presence of the plaque, which means that in the end stage of the disease, if you remove the plaque, they are still sitting there. And some mechanism probably already happening and induced across multiple cell type. They are still there, keep driving the disease progression. So if that is the case, we are very interested to understand what is the cellular coordinated network induced by amyloid plaque. And from lot of literature research, it's clearly that not only one cell type, but multiple cell type, they have very weird morphologies around the plaque, but not a cell far from the plaque. So we consider it's possible. It's a network sharing by multiple cell type, and they further propagate this pathology stress, the signaling out of this kind of microenvironments. So then we want to understand what's the molecule, what's the cell type involved in this network. And to understand these kind of questions, initially we have immunohistochemistry, and we use the 80 mouse model, which is a APB NRGF knocking model. So the conal section of the mouse brain from 3 to 18 months of age. So you can see the green, that is the GFAP reactive astrocyte. So that form already kind of green envelope around the white, that is amyloid plaque, and the very initial stage of the disease. So among the disease progression, those cellular networks actually spreading over. Is probably depending on the presence of the plaque and also propagating to the other brain regions. But then if we want to identify a cellular coordinated network, it's impossible to use HC. We need a global molecular profile with spatial information. So we use spatial transcriptomic. It's kind of a 2D RNA sequencing array. So in the old version of the spatial transcriptomic, there are 1007 spots on top of an array. So for each array, they have 200 millions of the oligos. And each oligo, they have polyT-tail and ID barcodes for each spot, the spatial localization. After we permeabilize the tissue, the mRNA will leak out from the cell and attach to the polyT-tail on the array. We do the in situ reverse transcription, and we collect all the cDNA afterwards. And for the library construction, then after sequencing, we can trace back the spatial localization of each molecule based on the ID barcodes. So for each spot, the yellow spot showing here, they will provide us the global transcriptomic profile of the corresponding unique tissue domain. Then the spatial resolution of each tissue domain, which is 100 micrometer in diameter of each spot. And the thickness of the tissue is 10 micrometer. So the spatial resolution is very compatible with our pathology amyloid plaque. That is actually in between 10 to 100 micrometer in diameter. So this is our experimental design. The middle section we use for the RNA sequencing, and we also collect the very two adjacent section from the cryo state we use for the immunostaining. So then if we have the amyloid plaque in either two adjacent sections, then we have very high confidence that the middle section, the corresponding spot, is collecting the transcriptomic profile in the cells in the vicinity of amyloid plaque. Because we want to link the transcriptomic profile to the abundance of the amyloid plaque, so we do the quantification of the A-beta based on the fluorescence of 6010. So the quantification is based on the amount of the A-beta. So you can see that like this spot, they have a very intensive fluorescence here that result in a much higher value after the quantification. And in this spot, they have diffuse plaque. Although they cover a huge area, but there is relative amount of the beta deposit in this spot. So eventually they give us a relative lower value after the quantification. Then based on this experimental design, we have for each spot, the full transcriptomic profile of in average 7,000 of the genes and the average corresponding level of the A-beta deposition of each spot. Then for this sandwich package, we carry it out on 2 genotype and 4 time points. So eventually we have more than 10,000 of the transcriptomic profile across 2 genotype and 4 time points. So the Disney plot, they can separate based on the genotype, time points, and the brand regions. So we download the Allen brand atlas and do the image alignment so you will know which region the spot coming from. So they can even separate the refined spatial localization of the spot in a somatic layer of the hippocampus. So like very small three group here in the red. This is the somatic layer of the dentate gyrus. And this one is the CA3 and this is CA1. So after those overview of the database, we have kind of confidence is biological meaningful. And we want to understand whether this database can cluster the gene co-expression network driven by certain biological factor. For example, if there's a landmark of the dentate gyrus, then that will only co-express and express, especially from the spot from the dentate gyrus. Then it's also possible that some genes, they're only showing and highly expressed in the presence of the amyloid plaque, then they might co-express and their expression level might depending on the level of the plaque. So to do this kind of a gene co-expression network analysis, we use a very well-established algorithm, the WGCNA. which has been shown in multiple publications to show they can identify gene co-expressed together. Normally they're functioning together. So more complex of your database is, then they will have higher chance you can find a refined network driven by a particular biological factor. So here we do it unbiasedly across our full database. So it's like more than 10,000 of the profile. We don't put any assumption for analysis. It could be driven by the age, genotype, brain region, or the level of A beta, or brain regions, or cell types. So to understand what's the biological driven of the co-expression of each network, we want to understand the differential expression of the network to the AD versus wild type mouse model. So we plot the differential gene expression of each network, whether that is up or down regulated in AD versus wild type, or whether that is up or down regulated under the A beta stress. So the plot showing here, that is the mean of the logbook change of the genes within each network. And the yellow spot is the result, the gene alteration from the 3 months. And the blue is the gene alteration and the 18 months. Then you can see that there is an outlier, the purple network, which is upregulated at the 18 months under the genotype axis and A beta axis. And but this network is kind of silent at the 3 months. So the yellow spot here is not too much change. And the other interesting network is the red network, which is upregulated at 3 months, but downregulated at 18 months in the A beta axis. So we particularly look into more detail of those two networks. We also do a lot of characterization to estimate what's the cellular composition of each network and which brain region they express. So eventually we name this purple network, which is composed by 57 of the genes, as plaque-induced genes network, the PICS network. And the red network is really enriched with a lot of myelin-related transcript. We call it oligodendrocyte network. So here I'm going to show you the detail of each genes within each network. The red is the oligodendrocyte. So they are upregulated in the plaque axis in the plaque at 3 months, but they are depleted at 18 months in this microenvironment. And the PICS is silent at 3 months, and most of the component of the PICS, they are upregulated in AD, especially in the plaque niche. So then I'm going to show you detail about the PICS. It's composed by 57 of the genes. So the plot showing here, the green indicate the strength of the connectivity score of the gene pairs of the PICS. The darker the color indicate the higher co-expression of the gene pairs within the selected transcriptomic profile. And the picture showing here is the transcriptomic profile selected only from the wild-type mice brain. And you can see it's not really co-expressed among all the gene pairs. They split into 3 clusters. The green cluster, they have very strong internal conductivity, and many of them, they are kind of well known. They are expressed by astrocyte. And the blue and the orange cluster, many of them are expressed by microglia. But this PICS network, when we start to look into the transcriptomic profile in the AD brain, so now I only select the AD transcriptomic profile with lowest A beta deposition. They already start to build on this kind of connectivity between the gene pairs and between the clusters. And if we gradually increase the amount of the A-beta, you can see that the connectivity score is going to be more and more stronger. So which indicate this network is gradually built up based on the amount of the A-beta deposition in each tissue domain. And especially this orange network, this orange cluster, they start to bring into the network, especially via the TRIM2 and TARO-BP, with their projection, their connectivity to the ApoE and the C1q ABC. And we are kind of surprised because this unbiased analysis, which actually point out the one of the most important molecule in Alzheimer's disease. And based on the gene ontology analysis, it indicates that this network is overactivation of the classical complement cascade. For example, they have C1q-ABC and C4-AB and this CLU. They also have a lot of downstream effector of the complement cascade. For example, lysosomal dysfunction, antigen processing and presentation, oxidative stress, immune response. So now we find a network which corresponding to the plaque deposition, and we know the mechanism, we know the molecule, and we also know what's the potential functioning mechanism of this network. But we don't know what's the cell which represent and express and driving this network. So we overlap our network together with previously identified marker gene of the disease-associated astrocyte and microglia. So it's clearly not only from one cell type, we also have some genes which is not identified as those kind of cell markers. So to get a real experiment and a single cell resolution, we incorporate the other orthogonal technology, the in situ sequencing, with the collaboration with CARTANA. The technology enables us to get a gene expression profile for more than 100 targets together on one section in microscope resolution. So then the picture showing here is the puncta of the PRAX1, which is the landmark of the dentate gyrus. And I'm going to show you the expression of the cell type markers. The red puncta is from the neuron D6 or synaptophysin. And the blue is from oligodendrocyte markers, and the green from astrocyte markers, and the yellow from microglia markers from those genes. And you can already see a cluster of green and yellow. And that's actually the place of amyloid plaque. So we can do the immunostaining after the in situ sequencing on top of the same section. So then we can try to use this technology to understand what the cell express our pigs in the mouse brain. We do this experiment in 2 wild type and 2 AD brains at 18 months of age. So first of all, we try to see whether the pigs, they are enriched in the plaque. And to our surprise, the 2 orthogonal technologies, they give us very highly consistent results. So the y-axis is the log change of the genes to the A-beta level based on the spatial transcriptomic. And the x-axis is the log-odds ratio, the enrichment of the genes in the ring 1 and the plaque based on the in situ sequencing analysis. So both of them, they all show that the pigs is significantly upregulated around the plaque. And we will try to do the cell typing of each genes within our pigs. For example, we start from looking at the green subcluster of the pigs. There are many bubbles shown here. The bigger the bubble indicate the higher log-odds ratio of the enrichment of that genes correlate with the cell type marker of certain cell type. So then you can see most of them are expressed majorly by the astrocyte. But the interesting stuff, you can see the ApoE normally expressed by astrocyte in wild type in 80 moles, but they start to express by microglia around the amyloid plaque. And it's also interesting for C4A and B, so it's normally expressed by astrocyte, but they start to express by oligodendrocyte in the 80 mouse, especially around the amyloid plaque. So this indicates that this microenvironment that trigger the activation of microglia or oligodendrocyte to start to express ApoE and C4Q and B involving this network. We also do the same for the blue and orange cluster. And it seems that the majority of them are expressed by microglia, but neuron astrocyte or oligodendrocyte are sometimes also involved. We also do the similar practice in the human brain. So not only the PICs, we also do the in situ sequencing for the oligo network. And the superior frontal gyrus of postmortem human brain tissue. So together we do the 220 targets, 380 and 3 control. And the image showing here is the white amyloid plaque is co-localized with the red, the neurons. They are mostly in the gray matters. And the blue is oligodendrocyte, mostly in the white matters. And the purple image is showing here is the expression of our PICs. It's co-localized with, it's more enriched with the plaque and the gray matters. And for the oligodendrocyte, during the cell typing, it's really purely from the oligo network is really purely from the oligodendrocyte. So that's why we name it as an olig network. So I make a short summary of the PICS network. So we consider it's a gradually built up network between especially microglia and astrocyte and the vicinity of amyloid plaque. So this is the mean Z-score of the gene expression level of the PICS at the 3 months of age in different conditions, for example, in the wild-type mice or 80 mouse brain, depending on different level of the A-beta. It's kind of silent at 3 months, but the PICS, they start to increase the expression based on the level of the A-beta deposition in the 80 brains. However, this response, they don't really have any unique regional specificity. So the more red, that is more upregulated and the plaque niche, and the blue is depletion and the plaque. And with the mesh that indicates it's not significant of up or down regulation. So at 3 months is kind of silent, but 18 months, as long as they show the significance that's highly overexpressed around the plaque, but kind of all over the brain. And for the oligodendrocyte network, it's kind of interesting. Their expression level at 3 months is during the mild A-beta deposition, their expression level is gradually go up. But if there are humongous A-beta deposition, such as plaque in the brain, so even at 3 months, this network, they disrupt, they reduce the expression level. And for the 18 months, the oligodendrocyte network in the mild A-beta deposition, they already go up compared to the wild type, but they gradually decrease when we increase the A-beta per microenvironment. So we consider the olig network as an early reaction to the mild A-beta deposition, but they eventually break down if there are severe A-beta deposition in the brain. And this reaction actually have the regional diversity. For example, if you look at the 3 months of age, so this network is highly enriched in the plaque, especially in the fiber tract thalamus and hypothalamus, where are the brain region enriched with the myelin and oligodendrocyte. And for the 18 months of age, that is not very clear about the depletions in certain brain regions. So it's a more like a early reaction in the oligodendrocyte enriched area that have higher response to the plaque. And so I would like to especially appreciate several collaborators. It's a humongous work across multiple colleges, especially Ashley and Mark. They are bioinformaticians and they carry out majority of the analysis of this paper. And many people from Bart De Strooper's lab and people from Image Core in VIB-KU Leuven. So the Spatial Transcriptomic, which we have the collaboration with the Wei-Ting Chen from Karolinska. And now the Spatial Transcriptomic has been bought by Transcriptomics. It's available, it's called like a text Visium slides. And work together with CARTANA is now also being bought by Transcriptomics as well. So they are also available from that company. And the mice is from Riken, and the human brain sample is from the Brain Bank. Yeah, it's now open for questions. Thank you.
Thank you so much for the beautiful talk. While we are waiting for the questions to pour in, I personally have a question because our lab is interested in like multiple sclerosis and then like a normal appearing white matter, that it's how far from, like we're always curious like how far from the lesion that is considered normal. So I guess like in your system, you have the chance to really see if like how far away from the plaque, like the oligo modules, like a genes early responses, like back to like a homeostatic states. Like, yeah, like so my question is like, have you looked into that?
You mean that, did I look into the homeostasis of, sorry?
Yeah, like from the plaque, the center of the plaque, I assume that surrounding the oligo will respond to the plaque, and then by upregulation of like the myelin genes. And then I wonder like how far, like maybe like 5 micron or 100 micron from the plaque, and then you no longer see this upregulation. I wonder, have you checked that?
Yes, so from the spatial transcriptomic, we do the analysis in a different way, but from the in situ sequencing, it's possible to understand the distance from the plaque. So here you can see that we drew the rings based on the distance from the plaque. The white is the 60-10 standing. So we expand the mask of this kind of amyloid plaque by 10 micrometer in radius as the ring one. And then we expand by 20 micrometer as a radius for ring 2, ring 3 to 4, 5. Yeah. So then oligodendrocyte, we see the enrichment of the C4A and C4B in oligodendrocyte, especially in ring 1, and that gradually decrease from the ring 1 to ring 5. So so actually, after ring 2 is already almost going to the background levels. So the enrichment of the C4B in oligodendrocyte is like this, but for the enrichment in the astrocyte is more like in ring 2 or ring 3. And the reaction of the microglia is more like within the ring 1. Yes, that's the result from the in situ sequencing. But for the spatial transcriptomic, unfortunately, we do the quantification in the other way around. So we don't have like those rings because those spots are physically fixed by their spatial localization. Yep.
Thank you.
Okay, we have a question from Nathan Pomper. Great talk and work. Based on your staining for PLP1 and MBP, it suggests that your work focused on myelinating oligodendrocytes. Did you see any interesting phenotypes with regard to OPCs or newly born oligodendrocytes? In many other disease models, OPCs demonstrate increased proliferation, whereas new oligodendrocytes degenerate.
It's a really nice question. So unfortunately, during our experimental design, we did not really focus on different lineage of oligodendrocyte. So eventually we don't have a reliable marker to indicate whether the OPC is involved in the PICS or the omics network. So I cannot provide any answer with confidence, but we just aware this kind of important question after we generate all the data. And there are several interesting papers recently. They also find the OPC, they start to go into senescence and the plaque niche. And they stop to go like differentiate into the malignity oligodendrocyte, and they instead they release inflammatory cytokines. And they become to involving this kind of inflammatory network together with the microglia around amyloid plaque. But it's kind of pity here that our project can answer the question about OPC. Yeah.
Okay, well, quickly, next question is from Brian. Amazing data in the presentation. What is the expression of the complement molecules in oligodendrocytes at later stage in your AD model, like your second point of analysis, 18 months?
Mm-hmm. Sorry, the question is whether there's, could you repeat again? Sorry.
Sure. What is the expression of the complement molecules in the oligodendrocytes at the later stage?
When?
When? I guess is about the species. What is the expression of the complement molecules?
What's the expression?
Mm-hmm.
So the complement, so the PICS, the expression level, they are silent at 3 months and also 6 months, but then they are dramatically increased between 6 and 12 months. And after 12 months, those PICS and complement molecules, they are kind of saturated after 12 months. And for their expression and the oligodendrocyte, the species, sorry. So the question is about whether they also express, how they express in the human oligodendrocyte?
I got a clarification. That's I meant the expression level. Like, yeah, yeah. I think it can like further maybe reach out to Wei-Ting to further address the question. For now, like we thank Wei-Ting for a very
Okay, no problem.